Hello, everyone. This is Adam Tabera with Psychedelic Insights, and I'm very excited to be joined by Ian McDonald, the CEO of Bright Minds Biosciences today. Take it away, Ian.
Hey Adam, how are you? Good to see you. Yeah, excited to get going today.
All right, awesome. So we're just gonna jump right into this and let's get started. So the first question is, where do you think the psychedelics industry is at right now?
I mean, in what regard? In terms of development, in terms of sort of, give me a little more context there.
So let's say in terms of like drug development and the like.
Yeah, so we've got a lot of active compounds that are going through clinical trials right now. I mean, those are primarily first generation compounds like LSD, psilocybin, DMT, 5-MeO DMT. But yeah, a tremendous amount of work's been done, not only preclinically now, but also dozens and dozens of ongoing clinical studies. So I think we're really at a inflection point here in terms of where the science has gone, and really moving from these early phase clinical trials into larger, more powered studies that will really give, I would say, regulators the confidence that these are truly transformational therapies. So exciting times for us, and with MAPS getting close to approval here with their quite strong data for MDMA and PTSD, I think that will certainly be the first, but I think the first of many.
I hear that. So, to build on that, what are your opinions on overall investor interest in the sector, availability of capital, potential for industry consolidation, M&A?
Yeah, I would say it's anemic right now. It's really not an issue that's just facing this little subsector of biotech, but it's an issue of capital availability that's faced more broadly by risk assets. And I mean, we've really seen a step change from the days of COVID, call it late 2020, and pretty much all of 2021. In December, you really saw risk assets sell off. Early stage biotech certainly fitting in that bucket of companies that aren't earning any cash flow and won't be earning any revenues for some time in the future. So not too much of a surprise given the macro headwinds of where the junior biotech market is, and within that the psychedelics market. You're really not seeing too much of a disparity between the psychedelic subsector and early stage junior biotech. So yeah, I would say it's challenging right now in terms of getting institutional interest and retail interest. There is still, I would say, relatively strong support from government bodies and grant funding, but the days of being able to raise capital freely and easily at compelling valuations are behind us for the moment.
I hear that. Next question. Bright Minds has a reputation of having a lot of bright minds, great scientists, and medicinal chemists working at the company. Can you give us some background information on the team at Bright Minds and highlight some notable accomplishments these individuals have achieved in the past?
Yeah, so when we started the company, we really took the view that while these first generation psychedelics were efficacious, they really lacked a lot of the drug-like characteristics for them to be truly embraced by the medical establishment, by not only patients, but physicians, payers, and large pharmaceutical companies. So what we set out to do was really use those first generation psychedelics as a launching point to develop compounds that had the characteristics we were hoping to achieve. So the first stage in that is identifying what the problems are. For that, you really need to take a detailed look at the literature, have a pretty solid foundation on drug development. And once we had sort of those key target areas of improvement that we wanted to focus on, we needed to assemble a team that had the requisite knowledge to not only design those molecules, but also test those molecules and understand how they work in the body and what receptors are important for what actions, how to create a drug that has a short half-life, for example, and to do that, we really set out to find the best in breed within each of these little call it sub-disciplines. So on the medicinal chemistry side, we teamed up with Alan Kozakowski, who is quite a famous professor. He's late in his career now, but he's spent decades and decades and decades, and quite literally wrote the book on a drug design for neuropsychopharmaceuticals, so active brain drugs. And Alan had spent quite a lot of time working on the same problems that we were working on here at Bright Minds, which is the specific receptors associated with the psychedelic action, which is the 5-HT2A. And his not only knowledge of that specific receptor, but his broad knowledge of the brain, developing drugs, not only for serotonergic targets, but Alan is somebody with I would say really grounded knowledge, not only in medicinal chemistry but also neuroscience. So he after he got his degree at Berkeley, he went over to Harvard to study under a Nobel Prize winner, E.J. Corey, who's really one of the, I would say, one of the leaders in the 20th century for neuroscience. There, he understood quite detailed aspects of the brain, and went on to have a subsequent career in academia for decades and decades, where he was, I would argue, one of the leaders in the world in terms of developing cocaine analogs. So he developed a compound called nocaine, which was a replacement therapy similar to methadone for opioids. That never made it onto the market, but showed tremendous promise. I think that was more of a regulatory issue than anything. And he also worked for a long time on PCP, or angel dust, as people would know it, and that works on the same system as ketamine. So that works on NMDA, which is another neurotransmitter, but he's had a lot of experience developing these compounds. We also had another very strong member of our founding team on the medicinal chemistry side, and that's Gideon Shapiro. So Gideon also went to Berkeley. He was about a decade younger than Alan, and instead of going the famous professor route, he went the industry route. So he actually started his career off at Sandoz, which, as many people I think will know, is the birthplace of LSD. Was actually working in a lab one floor directly below where Albert Hofmann first synthesized LSD, and he went on to run the chemistry division of their CNS group. So very, very strong background from that aspect. But also he's worked on NMDA and creating a second generation version of ketamine. That's where he spent his last decade or so, and was quite successful in that. So basically what he did there was create a next gen, call it ketamine 2.0. With that company, they had a couple of big pharma deals, one with Allergan, another one with Otsuka. And what we were doing at Bright Minds is effectively the same thing, but instead of the NMDA receptor or the NMDA receptors, we were focused on serotonergic targets, in particular the serotonin 2A receptor. So yeah, with those two guys on the medicinal chemistry side, we really needed somebody who was at the top of the field in terms of the biology side. So we recruited a gentleman by the name of John McCorvey, who I would regard as probably the number one guy in terms of cutting edge neuroscience and really understanding the signaling pathways that are associated with the 5-HT2A receptor and adjacent receptors, and really getting a detailed understanding of how those work in the body, the interplay between different receptors. So with, I would say, those three core members, we had what we needed to then design drugs and put them through a primary screen. As we moved on, as I think many people will know, when you're developing a novel drug, you need to first invent the drug. You need to then screen it in preclinical tests. You start off in test tubes, then move to animals. So we bolster our team to really suit the phase of development the company was in. We added in a gentleman by the name of Jean-Min Duan, who was heading DMPK, which is understanding how the drug works in the body, in both animals as well as people, understanding how long the drug lasts, how well it gets absorbed, how quickly it gets absorbed, how quickly it gets excreted, any toxic metabolites that are associated with it, and so on. So he was a very storied drug developer, drug designer, really working the translational aspect of bridging preclinical and clinical at the world's largest privately held pharmaceutical company called Boehringer Ingelheim. And we managed to get him, and he's been a quite invaluable asset to the team. And once we developed these drugs, we really moved out of the discovery phase and into development. So in order to reflect that, we made some changes to the team. We had all the molecules we needed, so medicinal chemistry wasn't something the company was focused on, but we needed to develop these wonderful compounds that we made. So last year we added in a new chief scientific officer by the name of Jan Peterson. Jan was in charge of one of the guys in charge of the neuroscience group at Lundbeck, which is quite a large European pharmaceutical company with a lot of experience in psychiatric drugs, as well as drugs that work on neurological targets. He's also the co-founder of a company called Acadia Pharmaceuticals, which has a drug that works on 5-HT2A, but instead of an agonist where you turn the receptor on, they had an antagonist which turns the receptor off, and that drug is marketed for psychosis symptoms. So tremendous amount of knowledge about not only the specific receptors we're working on, but how big pharma views compounds, what they're looking for, how to take a really interesting compound and develop that properly and go from preclinical all the way through to a marketed drug. We also added a new chief medical officer. Our new chief medical officer is a gentleman by the name of Mark Smith. Mark is a brilliant guy, trained at Yale, got his medical license. He's not only a doctor, but he's also a psychiatrist. So he spent a long time in both large pharma at companies like AstraZeneca, running their psychiatric program, as well as working in smaller biotech companies similar to ours. So I think with those key members, we're really well positioned to take these drugs from this preclinical state all the way through developing into true pharmaceuticals.
Yeah, sounds like you've assembled quite the team. Next question. What potential benefits does Bright Minds see in targeting both the 5-HT2A and the 5-HT2C receptors?
Okay, let's start with 5-HT2C. So 5-HT2C is, I should start off by saying we're creating agonists, so drugs that turn on the receptor both in 5-HT2A and 5-HT2C. When you turn on the 5-HT2C receptor, there are several benefits. First, I'd say first and foremost would be the impact on epilepsies. So you have a couple of marketed drugs that work on the same receptor, which have shown quite significant ability to reduce not only the seizure frequency but also the seizure duration. And those compounds have really been tremendously impactful within rare pediatric epilepsies, syndromes like Dravet syndrome. Also would have application for Lennox-Gastaut syndrome, and there's about 25 of these disorders that fall in that fall into I'd say a similar bucket. And these are generally quite rare disorders, which are not very well suited by the drugs that are on the market today. And 5-HT2C and a drug like ours could have tremendous application for that patient population. These compounds are also work quite well as antipsychotics. So we've done some preliminary test work with compounds in the series, and what we're seeing is the ability for these compounds to affect agitation. So typically, if you have a dementia patient, someone with late-stage dementia, they'll typically have hallucinations. They will typically start to get aggressive in the later phases of the disease, and that can lead to a lot of disruption within the household, which typically is the deciding factor for putting a loved one in a care home. And what our drugs seem to do, at least preclinically, is reduce that aggression, reduce the psychosis symptoms. So would really be, I would say, a step change in treating these late-stage dementia patients. Another third and final bucket that I'm going to touch on here with 5-HT2C is addictions broadly. So just touching on the pharmacology here, 5-HT2C is co-located with dopamine. Dopamine is primarily associated with the reward system, and if you have an addiction, whether it's smoking, whether it's a gambling addiction, whether it's social media addiction, typically it will affect the dopamine pathway. And our agonist, our 5-HT2C agonist, since it's co-located with those dopamine receptors, blunts the response. So let's say I have a social media addiction and I want to keep going on Instagram. You'll typically have a rush, a dopamine rush. When you post a new photo, you get new likes, whatever it is. What our drug does is dampens that dopamine response. So really works to take the craving away from opening the app 10 times, or if you have a smoking addiction, from lighting up another cigarette or puffing on a vape. And we've tested this in a myriad of different disorders. You could have application in this from a disorder like binge eating disorder, where it's really a food addiction. We've tested in that, seen very significant reductions in self-administration. We've tested in nicotine, we've tested in opioids, and seeing some pretty substantial benefit there. There are other diseases you could use this in. Prader-Willi syndrome would be an example. That's something we're looking at, which is a disease with developmentally disabled children who generally have issues with overeating as well as aggression. So we think our compound could work for a couple different hallmarks of that disease. And then on 5-HT2A, I think this is pretty well documented. I don't know how in depth I need to get to on this, but basically what 5-HT2A does in a very simplified explanation is it allows your brain to function normally, and instead of getting caught in a feedback loop, typically a negative feedback loop between a couple parts of your brain, you have effectively a reset effect where your brain is allowed to act like a normal functioning brain, at least for a period of time. Typically, if you have a true circuit disorder, like a treatment-resistant depression or a moderate to severe form of depression or an addiction, you'll generally drift back into dysfunction after a period of time following these medicines, but it will give you a benefit for some period of time.
Got you. A question about the 5-HT2C. So, saying you were targeting addiction, right?
Yeah.
So if you're dampening the dopamine response, is that something that would translate into anything that they would be doing? For example, like not just addictive behavior, or let's say just like getting work done at home, and like you want a good you want a positive dopamine response from that as like the reward for your body for doing something good, right? So
yeah, I mean, I think it's important to say, like, okay, let's take a methamphetamine addict, somebody who's using a large amount of crystal meth or other amphetamines. Typically, they will burn through those dopamine receptors, and after a while, they're not really getting much of a positive response, if anything. So, I think it's really important to understand that these receptors can be impacted by these drugs, and really, those people won't really have much dopamine going around when they're doing normal things. So I think it's really important to take away the core driver of that dysfunction, which is generally the addiction.
Understood. Next question. In the past, you had mentioned that Big Pharma was showing interest in the company and its pipeline. This makes sense considering you're working on NCs that have composition of matter patents. Is Big Pharma still showing interest in your pipeline?
Yeah, definitely. So I'm headed along with a couple of other members of our team to one of the largest partnering events in the pharmaceutical industry in a week or two. And I think we have over 35 meetings scheduled already. A lot of those with household names. So there's certainly a lot of interest, and that seems to be continuing. So yeah, it's been quite positive.
What do you think is holding Big Pharma back from kind of jumping in earnest? It seems like there's a lot of waiting on the sideline right now.
Yeah, I think there's a couple different issues. A lot of the drugs that we've seen today have been, I would say, either not patentable or pretty weak patent protection, which is not an ideal scenario for a company that's looking to invest heavily in that drug and get a return on that. So I think that's been a part of it. Outside of that, yeah, there's been some activity. Companies like Otsuka have dipped their toes in with arketamine. They've partnered with Gideon's company, which has a version of ketamine. There have been a couple other companies that are that I think have been very, very close to doing something. But yeah, there's still a lot of wait and see. I think we're going to need to see a little bit more clinical data. Some have apprehension on the liability aspect of a psychedelic drug. Again, this isn't everybody. Others have apprehension on the treatment model, but I think largely there's a lot of interest in these compounds, and as these molecules progress further through the development pathway, then there'll be more interest. Most companies usually look to see clinical proof of concept before doing a deal. There's other companies that are more, I would say, comfortable with the mechanism. Otsuka would be an example. that are doing deals earlier, but that would be an exception rather than the rule of clinical efficacy data, at least in a small patient population.
Understood. Next question. Bright Minds was one of the first companies in the space to look at novel drugs targeting the 5-HT2A receptor. Nowadays, many companies are doing this work. What separates your company's 5-HT2A agonists from others in the space?
We haven't seen anybody with. I'm going to be completely frank here. We haven't seen anybody with compounds that are anywhere close to ours in terms of selectivity, have the attributes of a high Emax, which is something we were looking for, quite a more of a full agonist as opposed to a partial agonist, and kind of hit that sweet spot of short acting. So yeah, it'll be interesting to see some of these other companies and exactly what they're targeting, and see some of that data. But we've been thrilled with the compounds that our discovery efforts have yielded.
Got you. Do you have any ideas what might have caused the stock to soar 400% in August of 2022?
Yeah, I think that was primarily retail. So you had quite a large, influx of retail buying over the course of a few days. I mean, you see that in the space. It's not only in psychedelics. Like you took a look at Embernick today. It was up. It was like 2x on, and I think they traded like 4x their shares outstanding in one day. So that's not limited to psychedelic stocks. You're seeing that kind of across the board with these beat down biotechs. So yeah, things can change pretty quickly in biotech land, and I think that was an example of that.
All right, and so to build on the last question I asked before, this is basically BMB 202 is described as a selective 5-HT2A fast on, fast off compound with a high Cmax and a short plasma half-life. Can you explain to the viewers what this means exactly?
Yeah, sure. So Cmax just means concentration max when you have the most drug in your system. And what we're trying to do, I don't know if you can see me here, but basically you want the drug to hit your blood really quickly, get in your bloodstream, get onto the receptor, and reach a, call it threshold limit, where you have this brain reset effect. And then after that's done, you want the drug to be relatively rapidly metabolized. So it's not sitting on the receptor for a long time like LSD, which, while like fun in a lot of instances, isn't I would say ideal from a rollout standpoint, from a patient access standpoint. Just too long to be sitting around, and certainly it's too long to be sitting around under supervision of a medical professional who's earning $150-$200 an hour. So what we look to do is have a drug that, again, hit you really quickly, hit you hard, and then was rapidly metabolized. So that's what we did with BMB 202, and a lot of other compounds within our portfolio.
So specifically for BMB 202, what would be the onset? How quickly would the onset be, and how long would it? Is it like intravenous? If you give us any more information on this.
Yeah, sure. So, I mean, you could formulate this a bunch of different ways. You could do IV. I would say subcutaneous would be probably the best the best delivery form. It hits you very quickly. This is something that would be potentially administered at home, or would have the flexibility to with that delivery method. And this is the same way that people take diabetes insulin shots. So yeah, we'd be looking at a subcutaneous delivery method, and the drug would hit you very quickly within minutes. And I would expect the I would expect the psychedelic experience to be under 2 hours.
Under 2 hours. And kind of what, I mean, it's hard to, I guess, have an equivalent here, but like what type of, how far are you trying to push the psychedelic experience with BMB 202? Is supposed to be like, let's say, what would be like a eighth of shrooms? I know it's like hard to compare this stuff, but like kind of what are you targeting?
No, it's we're gonna be doing a dose range finding study. So we're gonna test out a bunch of different doses, but our drug BMB 202 is, call it, roughly 2 times as potent as psilocybin. We have other drugs in our portfolio which are, call it, 10, 20 times as potent, but BMB 202 is roughly twice as potent. So eighth of shrooms, what would that be? 3.5 grams, 25, 30, probably be 30 milligrams. So our drug would be like 15 milligrams to have the same experience, somewhere in there. Actually, a little bit less, because if you're taking psilocybin orally, it sort of takes away some of that effect, because it's entering the bloodstream slower. So yeah, it'd probably be like 10 to 12 milligrams.
And you're saying it's topping out about a 2-hour experience.
Yeah.
That seems extremely significant, considering what like all these other companies are trying to do with shorter acting molecules or shorter acting new chemical entities.
Yeah, it's not easy. I mean, it's not easy. It's really, really difficult to do. It's really difficult to get both a clean compound from a pharmacology standpoint and a short acting compound. So as far as we know, we haven't seen anything like this molecule, other than a couple others ones we created, which are effectively backups to this. But yeah, it's quite difficult to get that short acting nature in scaffolds apart from tryptamines, which are quite dirty in nature.
I got you. And basically what you said in terms of like you compared LSD, right? Saying how like, so if you're sitting with a therapist, let's say it's a 6 to 8 hour experience, that's messy, to say the least. And kind of that's I think I've been outspoken in terms of how I feel about how things will go. I'm happy that Compass is going to be first to market. That's great. It's going to be great for the sector, but I'm just not very optimistic in terms of how efficient that's all going to be once it's rolled out. So I think kind of compounds like this are going to be what actually really what really starts to kind of create the foundation for kind of how we will be administering kind of psychedelic drugs, psychedelic therapy.
Yeah, totally. I think we completely agree. I think that's going to be the future of psychedelic therapy. It's going to be these shorter acting compounds, that pharmaceutical companies can really get behind and contribute significant capital to, and really roll out knowing that they're going to get a return on that investment.
Got you. Next question: is Bright Minds going after treatment-resistant depression or MDD with BMB 202? Will trial participants remain on SSRIs or SNRIs?
Probably not with regard to SSRIs and SNRIs. You have a heightened risk of serotonin syndrome there, which I'm sure you know. But potentially you could run these in conjunction with those. Our compounds have, like BMB 202, for example, we've run toxicity studies on this, and we're seeing a 200-fold window of any observed negative event, and a NOEL of 200x on this.
Wow. So
incredibly safe, and I think that's pretty crazy looking at the selectivity and the fact that this is a full agonist. Whereas you see compounds like, call it psilocybin, LSD, topping out around the low 80s. This is a true full agonist where you're looking at finding Emax of, call it, somewhere in the high 90s. So really interesting from that standpoint.
So because it's a full agonist, would this kind of shape the psychedelic experience any differently than, let's say, a partial agonist?
Yeah, it should be more intense. I should rephrase that. It should be a stronger experience. And typically what we've seen, so this is why we want to design a full agonist, or why we think a full agonist is important. It looks like, and this hasn't been published yet, but it looks like there's a correlation between strength of psychedelic experience, i.e. Emax, and efficacy, as well as duration of that efficacious effect. So conceptually, this compound should have better or could have better efficacy and could last longer. And that needs to be borne out by science, clinical studies. But that's our hope, and that's our feeling.
That's extremely exciting.
Yeah,
so,
and there really haven't been too many full agonists in clinical development. You're really looking at the tryptamines. Anything in the tryptamine space is tough to get anywhere outside of the 80s. I don't know if I've seen anything like above 85, and we've literally created like hundreds of these tryptamines. And then looking at ergotamines, kind of similar, like LSD, kind of similar. There's a couple other chemical scaffolds there with ibogaine type compounds. With that chemotype, you can get full agonists, but there's not too many. There's not too many chemotypes that you can do that with.
That's interesting. All right, next question. When we look at your pipeline, we notice the company is looking at non-hallucinogenic 5-HT2A agonists. We have noticed companies looking at non-hallucinogenic psychoplastogens for their potential antidepressant, anxiolytic, and pro-cognitive effects. Does Brightmind see any potential for these drugs outside of these indications?
We don't know yet. It's relative to you kind of have two different ways to attack this, or two different theses. You have the Cmax hypothesis, which is that short acting, hit you quickly, hit you hard, true psychedelic experience, and you have that brain reset effect. That's, I would say, proven or close to proven at this point with all the clinical trials that have gone on with psilocybin, LSD, etc. And then you have another thesis with this, call it psychoplastogen approach, which is a subhallucinogenic dose, or a compound that just doesn't have that intrinsic efficacy that gets you above the threshold limit to produce a psychedelic experience. So that is still an open question. And what we had, which I don't think the field has really seen yet, are truly selective compounds to test that hypothesis. I think a lot of the other compounds that have been used in this have been dirtier type compounds, which hit a number of different receptors. So it's difficult to say what receptors would be at play with regard to the therapeutic action. So what we have are truly selective 5-HT2A compounds, where we can really answer that question of, is it the 5-HT2A receptor, the psychedelic receptor, that's responsible for this therapeutic action, or is it something else? And if it's something else, is that something that's already been proven? So yeah, we've got a lot of interesting tools to test that hypothesis, but it's certainly not proven, and I wouldn't say it's anywhere close to the certainty of the Cmax, the true psychedelic experience hypothesis.
Understood. So in regards to the Cmax thesis, wouldn't DMT just fit perfectly into kind of what you're talking about, because of essentially that peak breakthrough psychedelic experience, short acting, and kind of just in and out?
Yeah, DMT is pretty good. I don't think it's perfect. I think there's room for improvement, but it's a good compound, and I think I like DMT. It's
Uh,
good.
could you talk about some of the room for improvement there?
Yeah, sure. It doesn't have a ton of selectivity. You hitting different serotonergic receptors. Hitting different serotonergic receptors can lead to side effects. You have it being a tryptamine. It's quite unselective. You have 5-HT2B liability there, which I think has been a lot of people have talked about, including us, and something that FDA is quite sensitive about. You also have activity at some of these other serotonergic targets, which can lead to anxiety in the come up. So
a lot of anxiety on the comic, actually.
yeah,
Your heart races.
Totally. So I think that's something that's really unique, especially about BMB 202, where you really don't have that. So it's a lot. I don't know if you've ever tried a highly selective 5-HT2A compound, but it's way cleaner. It feels way cleaner. It feels much less anxiogenic on the come up. So you're not sort of riddled with anxiety like you are with certainly with like a 5-MeO, where you have some dysphoria and kind of challenging come up. So yeah, I think that would be one area of improvement for DMT. Route of administration, I mean, you can do it intravenously, which I guess is fine. Yeah, there's you also have, I believe you have some dopaminergic activity with DMT, which could be a potential liability in terms of regulators' view on it. I'd have to take a look at more at the pharmacology of DMT, but there's certainly some points there.
So now you have me curious in regards to BMB-202. So would the psychedelic space that you're in be kind of similar to like psilocybin, similar to, let's say, DMT, similar to LSD? Like where does it compare?
You're talking about like in terms of like the chemical scaffold, in terms of the structure?
Yes.
It would be closer to like a mescaline.
Oh, okay. Interesting. So it would be more dopaminergic?
No. I mean, we don't really hit dopamine. We don't hit dopamine at all. It's like it's insanely clean to 5-HT2A.
That's really interesting.
So yeah, it's chemically, I would say, out of the known natural compounds, it most closely resembles mescaline.
All right, cool. Sorry, that was just my own curiosity getting the best of me. Next question. So this I found really interesting. So I'd love to hear more about BMB Dash XXX. You mentioned on the website that it's 10 times more potent than psilocin. That seems extremely significant. I'd love to just hear more about it from you.
Oh, yeah, that's a cool one.
Yeah, my eyes lit up when I read about that.
Yeah. I gotta check and see which one that is. It's either one of two. We have two that would fit that bill. Does it say what chemotype it is on there?
So mixed 5-HT2A/2C compound, tenfold more potent than psilocin at 5-HT2A, ADM EPK profiling.
Okay. Yeah. So yeah, we've got a couple compounds. I don't think we've announced yet because we have a couple different compounds that have a couple different chemotypes, which look really interesting. There. Highly potent. Again, one of them is more rapid acting than the other, and highly selective for those two receptors. And it's more of a mixed agonist profile, meaning that it hits both the 5-HT2A and the 5-HT2C, which is interesting. It's, I would say, more like your traditional tryptamines in terms of hitting both of those receptors. But again, you wouldn't have activity at the other serotonergic targets, which can lead to some of those side effects that we talked about before, like nausea on the come up, etc. So yeah, that's a really interesting one. And again, you would like arguably need like 2 milligrams of this stuff, like barely anything.
Yeah, that's pretty. I mean, that's significant as hell.
Yeah.
Okay, so next question: what do you see as the biggest near-term catalyst to really drive this sector forward? I feel like you touched on this before by mentioning maps, but if you can build on that a little more.
Yeah, I mean, if I'm going to be completely honest, it's going to be macro driven. I think it's going to be, yeah, you're going to have some moves. You're going to have some moves in the space, and there's going to be some announcements. I think the space isn't really going to move until you have a change in the macro. Probably need rates to start dropping again. And need risk assets to come back in vogue, and once that happens, you'll probably see a reflation of these assets. And I don't know how long that's going to be away. Hopefully not too long. But that absent, yeah, you could see some moves with announcements. But I would say the space has seen a number of announcements that have been relatively positive, but haven't really seen the share price appreciation that you would expect, given the materiality of those events. So yeah, I think we really need to see that paradigm change.
Got you. Last one. Can you talk about your cash runway and how are you guys approaching raising capital? What does that look like? Kind of give us spill the tea.
Yeah, sure. So we've got, this is the last reported financials, about 10 million in the bank. And yeah, we can last another couple years, basically ride out this bear market here. See the other side of that. We have a very long list of compounds. Those compounds we can potentially monetize and use the proceeds of that to fund our development. We could also potentially license some of those compounds and participate in the upside of those compounds, but not be paying for development. That's a pretty typical model you see in biotech. So yeah, a few different ways to play it, but yeah, it's there's no way around it. I mean, it's a challenging environment. I know we can raise money, but it's a question of, do you want to raise money at dilutive levels? So yeah, that's sort of where we're at. I don't think we've touched on, just quickly, I don't think we touched on BMB 201, which is one of these compounds that fits into the low to no psychedelic response. This is a tryptamine that we created. It's a psilocin analog. And what we've seen here is this is a really unique compound in terms of its biased agonism. So this is the compound that we would expect to have very little, if any, tolerance buildup. So this could be potentially a chronic use therapy. We've dialed out 5-HT2B completely from this compound, so you don't have the issues with chronic administration and activating 5-HT2B, which is a huge liability in terms of the FDA's eyes, and a compound that isn't going to, like when you do shrooms, for example, you need to, if you do, let's say 2 grams today and 2 grams tomorrow, those 2 grams tomorrow aren't going to hit you the same as they are
not even close
day one. And that's due to this tolerance issue. So it looks like we've addressed that in this compound, which would mean that you can take this in a chronic potentially daily basis and not see an issue of needing to increase dose. And this compound, this is pretty new data that we've received, but it looks like this compound is pretty close to approaching as close to BDNF in terms of its ability to stimulate neuroplasticity and its ability to stimulate neuron and dendrite outgrowth. So really interesting from that perspective. So that's a compound that we're really encouraged by.
It's BMB 201.
Yeah, BMB 201.
That's so, like, in general, I find that I don't even like doing shrooms or psilocybin anymore. I prefer, like, for example, either prodrugs of psilocin or analogs of psilocin, because there's just a lot less anxiety on the come up. It feels
Yeah,
like a more pure psychedelic experience,
yeah,
and it's just more better tolerated by my body. And I
yeah.
think that, like, the vast majority out there haven't experienced it yet, but once they do, I think there's going to be a strong preference.
I think so too. Yeah, I think so too. A lot of those natural alkaloids in the mushroom really don't sit well with this,
They
the
get really
protein. So
hairy sometimes.
yeah, totally.
So yeah, so BMB 201. I'm sorry, I didn't see that on the website. That's why I didn't mention it. But is there anything else you want to say about it?
No, I mean we've run a number of preclinical studies on it, and it looks like it's going to be going to potentially be quite impactful in its ability to work on some of these neuropsychiatric conditions. I mean, with something like that, you could potentially use that for like a cluster headache, for example, or a lot of other applications would be.
Even like pain management, right?
Yeah, pain management as well. Yeah, we're actually working with uh, National Institutes of Health, testing that compound out in various pain models to see how that works. So stay tuned on that.
And like, what would the duration be on that on BMB 201?
Would be similar to, I would say similar to psilocin, probably 6 hours ish, 4 to 6 hours, something like that. But that's a compound. This is a compound that we've really toned down, almost eliminated the psychoactive component. So if you feel it, it would probably be equivalent to, I'm just guessing here, it would probably be equivalent to like a third of a gram, half a gram
Gotcha.
of mushrooms.
That's awesome. That's really, so when can we
Yeah.
expect to find out more about BMB 201?
We're working on a paper on that compound right now. So yeah, there should be. I think that's pretty close to wrapping up. So hopefully we'll have that released in the next couple of months here.
Awesome. I'd love to have you back on the show to talk about it when the time comes.
Amazing.
And I got the last question here. Is based on our knowledge, it's been a while since anyone from the team at Bright Minds has done a video interview. Why now?
We've really just been keeping our head down and focusing on the science. I don't think it really hasn't been a market that's been really conducive to conducive to publicity, and yeah, we've really been focusing our attention on advancing the compounds, on these discussions with pharmaceutical companies, and yeah, it's been a pretty exciting time in terms of the work in the lab and the science. And we like you guys. We like the program. So.
Oh, thank you so much. So hopefully we can expect to hear a lot more from Bright Minds coming in the near future. And once again, you're more than invited to come back on here whenever you'd like. I want to thank everyone for tuning in and watching, and I want to thank Ian McDonald for coming on the show. Thank you very much. And if you want, you can give like any closing statements to the viewers, Ian.
Yeah, thanks for watching. And if you have any questions, just hit us up.
All right, awesome. Thank you guys so much, and have a great day. This is Psychedelic Insights. I hope you guys enjoyed that conversation as much as I did. If you guys could please like and subscribe, I would really appreciate it. It helps with promoting these videos throughout the YouTube algorithm. So please drop a like, subscribe if you like the content that you're seeing. There's a lot more to come, and stay tuned for more just psychedelic videos in general and interviews as well.